Hello students, so coming back to chapter number 2, that is sexual reproduction in flowering plant. In this video class we will discuss about post-fertilization event in plant. And in post-fertilization event we will discuss about the formation of endosperm, formation of embryo, formation of seed and formation of fruit. But in this video class we will study only two topics, that is endosperm and embryo, and for fruit and seed we will discuss in another class. Okay, so first come to endosperm. We know that this endosperm is a result of a triple fusion. We also know that this endosperm have diploid number of chromosome. Not only that, this endosperm is actually a nutritive tissue which will provide nourishment to the developing embryo. Okay, this we have discussed in earlier class. Okay, now here we are going to discuss one important point related to endosperm and embryo, that is this particular endosperm will develop earlier than embryo, and the question is why. Okay, so to understand this question, let us go back to double fertilization. Okay, let us remind double fertilization. In double fertilization, what happened? Two fertilization event takes place. Okay, and after the end of the first fertilization, this embryo is going to be produced, and this endosperm is produced only after second fertilization. Okay, so definitely from this point of view you can say that this embryo should develop earlier than endosperm, but in reality endosperm develop earlier than embryo, and the question is why. Okay, so this endosperm develop earlier than embryo because this endosperm is the only tissue that is going to provide nourishment for the developing embryo. Because of that, embryo, which is the result of first fertilization, cannot develop earlier than endosperm, which is the result of second fertilization. Because of this, definitely endosperm will develop earlier than embryo. Okay, got the point. Next, come to the types of endosperm. Here we will discuss a particular type of endosperm known as nuclear endosperm. Okay, and to understand about the development of nuclear endosperm, a simple diagram is given by the side. So during endosperm development, what happened? The primary endosperm nucleus of the central cell will divide repeatedly, and this nuclear division will not be immediately followed by the wall formation. So definitely the endosperm will be multinucleated. Okay, and such multinucleated endosperm are known as nuclear endosperm. That is, nuclear endosperm are multinucleated endosperm. Okay, and in this nuclear endosperm, what can happen? Slowly, wall formation can take place around this nucleus, okay? So that this multinucleated endosperm can become what? Multicellular endosperm. That is, nuclear endosperm can be transformed into what? Cellular endosperm, okay? Only after the formation of wall, okay? But in some plant, what can happen? This transformation may be incomplete. That is, in a particular endosperm, some portion will remain as nuclear endosperm and some portion will be transformed into what? Cellular endosperm. Okay, and this particular condition is found in coconut. That is, the coconut water that we drink, it represents what? Nuclear endosperm, and the white portion that we eat, it is known as kernel. Okay, and this kernel represent cellular endosperm. Okay, so this is something about endosperm. Next, come to embryo. We know that this embryo developed from zygote after obtaining nourishment from the endosperm. So after formation of the zygote will definitely remain in dormant state. That is, it will remain in inactive form for some time. Okay, so question is why. So this zygote remain in dormant state for some time because the zygote waits for the development of endosperm, because this endosperm is going to provide nourishment to the developing embryo. Okay, because of this the zygote remain in dormant state for some time. Okay, now let us discuss about the development of embryo in dicot plant. So in dicot plant, the zygote divide into two unequal cells. One is called suspensor cell and other is called embryo cell. And here the suspensor cell will divide repeatedly to form a long filamentous of cell. And on other side, the embryo cell will undergo two vertical division, which is followed by one transverse division to form two layers of cell. Okay, and here one layer of cell is called epibase cell and other layer of cell is called hypobase cell. And here the hypobasal will develop, the hypobasal region will develop into radicle, and epibasal region will develop into plumule and cotyledon. That is, hypobasal and epibasal, they will develop into the main part of the mature embryo, that is cotyledon, plumule and radicle. Then these hypobasal and epibasal cell will divide repeatedly to form a globular embryo, which is somewhat spherical in shape. Okay. Then more growth will take place on the lateral side of this globular embryo to form the heart-shaped embryo. Like this, more growth will again take place on this lateral side and it differentiates to form cotyledon. So like this, the zygote develop into mature embryo. So in dicote, what happen? Mature embryo will contain two cotyledon, one plumule and one radicle. Like this, from a zygote, a mature dicote embryo will be produced. Next, come to development of monocote embryo. In case of monocote, the zygote also divides to form proembryo, then globular embryo, then heart-shaped embryo, and then mature embryo. Here, the developmental process is same with that of dicote embryo, but the only difference is that in case of monocote, only one cotyledon will develop, and that developed cotyledon is known as scutellum, and the undeveloped cotyledon is known as epiblast. Okay, so this is about the development of monocote embryo. Now come to the structure of dicote embryo. This dicote embryo, it consists of an embryonal axis with two cotyledon, and the embryonal axis has two ends. One is called plumule and one is called radicle. And the portion of the embryonal axis above the level of cotyledon, it is known as epicotyle, and this epicotyle will terminate with the plumules. On other end, the portion of the embryonal axis below the level of cotyledon, it is known as hypocotyle, and this hypocotyle will terminate with the radicle, and this radicle will be covered with a root cap. So this is about the structure of dicote embryo. Now come to monocote embryo. In case of monocote embryo, there is a single cotyledon, and that single cotyledon is known as scutellum. And there is also another structure known as epiblast, and actually this epiblast represent the undeveloped cotyledon. So in a monocote embryo, there will be a single cotyledon known as scutellum. In addition to this single cotyledon, there is also an embryonal axis, and this axis will definitely have two ends. One is called plumule and one is called radicle, and these two ends are covered or are supported by a protective covering, and that protective covering which is present above the level of shoot or above the level of plumule is known as chorioptile, and the protective sheath which is present above the level of radicle is known as choriorhiza. Okay, so this is about the structure of monocote embryo. Like this, we have come to the end of this class. Okay, and before we wind up, let me remind you what we have learned in this class. So today in this class, we have learned about endosperm embryo, about the structure of dicote and monocote embryo. Okay, and hope you can understand all the point, and thank you very much for your attention.